Hello everyone, I am Tommy. Welcome back to the series of the metabolism. Today I'm going to start the fat metabolism. In concrete, in this video, I will explain to you the fatty acid synthesis. So let's begin. Okay, so we start with glucose outside the cell. As always, do you remember glucose from glycolysis? This is a recap from glycolysis to pyruvate. Pyruvate gets inside the mitochondrion, acetyl-CoA, and then we get the Krebs cycle. Do you remember that? Citrate to isocitrate to alpha-ketoglutarate to succinyl-CoA to succinate, fumarate, malate, and oxaloacetate. This is the Krebs cycle. So for fatty acid synthesis to occur, we need high amounts of citrate. Why is that? Because when we have already too much ATP, this enzyme, the isocitrate dehydrogenase that converts the isocitrate into alpha-ketoglutarate, is inhibited by ATP. So we are stuck here in the cycle. We get acetyl-CoA, citrate, and isocitrate. But isocitrate can no longer be converted into alpha-ketoglutarate when we have high amounts of ATP. So, as this step is reversible, isocitrate is then converted back into citrate. Citrate can get outside of the mitochondrion, and a special enzyme will wait for citrate outside. Citrate lyase will split the citrate in two molecules. One is one we already know from the Krebs cycle, the oxaloacetate, which is 4 carbons. Oxaloacetate that can be converted into malate, and malate can be converted into pyruvate through the malic enzyme using NADP positive and creating NADPH. This is reducing power that can be used against free radicals, for example. Anyway, citrate lyase also creates acetyl-CoA using a CoA enzyme, of course, in the process. So we get acetyl-CoA, and then a special enzyme called acetyl-CoA carboxylase creates malonyl-CoA. Okay, so what is that? This enzyme is very important, and it depends on biotin, which is vitamin B7 or B8, depending on the literature. This enzyme is adding a carbon to the acetyl-CoA. Is inhibited by long-chain fatty acids. Why? Because indeed malonyl-CoA will be an ingredient, if we can say that, to create new fatty acids. Indeed, that makes sense, because if we already have fatty acids, there is no point in creating more. And then we have citrate as a stimulant to this enzyme, because if we have a lot of citrate, it means that isocitrate dehydrogenase has been shut down because we already have high quantities of ATP. Of course, to add carbon, we use the CO2. And here we have the explanation in depth. Basically, this enzyme has two forms, the dimeric and the polymerized. The dimeric is inactive and has phosphates, and the polymerized is the active form. So here, PPP and insulin stimulates phosphoprotein phosphatases, and phosphoprotein phosphatases steal the phosphates from the dimeric form, and they create the polymerized active form. So insulin is stimulating this process to happen. Insulin is stimulating indeed fatty acid synthesis. On the other hand, glucagon stimulates the protein kinase A that that adds the phosphates to the polymerized form, creating the dimeric form, which is inactive. So glucagon is inhibiting this activation, is actually inactivating this enzyme. This is the fatty acid synthase, which has a cysteine group and an ACP group, acyl carrier protein. These two groups are binded to the fatty acid synthase, and various reactions will happen between the two, which we're going to see them now as we approach this first reaction, which is the incorporation of acetyl-CoA into this enzyme. We use the acetyl transacylase. Okay, what does that mean? Acetyl, okay, transacylase. That's transferring an acyl group to where? To the acyl carrier protein. So acetyl-CoA releases the CoA and the acetyl group gets bind to the acyl carrier protein. Then we have the acetyl transacylase. The acyl transacylase basically gets this acyl group and transfers it to the cysteine, as you can see here, from the ACP terminal to the cysteine terminal. Okay, so now we have this complex here, and we add to it the malonyl-CoA. Do you remember we synthesized that from acetyl-CoA? Malonyl-CoA is added using malonyl transacylase. Malonyl transacylase, okay. Acetyl transacylase. Malonyl is 3 carbons. And yes, we are adding that to the ACP terminal. So we get the 2 carbons from the acetyl-CoA and the 3 carbons from the malonyl-CoA here. Then a very interesting enzyme starts to play. The acyl malonyl ACP condensing enzyme. Okay. What is doing this enzyme? The acyl, okay, this one, to where? Malonyl, okay, so this goes here, because malonyl on the ACP, acyl, malonyl, ACP condensing enzyme. So we are basically condensing these two into this terminal. And in the process, we lose 1 carbon in form of CO2. So the result is this one, as you can see here. And by the way, I am putting this double bond with oxygen here, because then you will understand better why. And here I put the beta with the oxygen group, because this enzyme we are going to use is the beta ketoacyl ACP reductase. Because you have to know that here on the beta carbon, because this is carbon number 1 alpha beta, on the beta carbon we have the keto group, and this enzyme is reducing that. So using NADPH, which we created, do you remember from where? From the malic enzyme that converts malate to pyruvate, from the malic enzyme. And as well, if you remember from the video of the pentose phosphate pathway, we create NADPH as well. We're going to use NADPH now in this reaction, because we are going to reduce this double bond here and get rid of it. As you can see, we continue and boom, we added hydrogens. Now we have the 3 hydroxyacyl dehydratase. Okay, what does it mean? 3 is the compound number 3. Hydroxyacyl is the hydroxy group on the acyl carrier protein terminal. Dehydratase is getting rid of water. So we're basically getting rid of these with another hydrogen here, and we create a double bond. As we can see, we create a double bond. And then this complex here with the double bond, a carbon, another carbon, and we call it enoyl. This is an enoyl group. Oxygen, carbon, carbon, double bond, carbon, carbon, whatever. Enoyl ACP reductase. Okay, reductase, we're going to reduce the enoyl on the ACP terminal. Using what to reduce? We used the NADPH. Okay, so we basically get rid of the double bond here. And this is the final result. This is actually a 4-carbon fatty acid, which this is a butyric acid, because if we use thioesterase, we can cut this bond to the ACP carrier protein and we release the fatty acid, and this is called the butyric acid. But to continue making the fatty acid longer, we can follow this arrow and we can repeat the steps. We can use an alkyl transacylase and move those 4 carbons to the cysteine group. Then we can add another malonyl CoA and using the malonyl transacylase to put it here. And then using the acyl malonyl ACP condensing enzyme, do you remember? We condense these 2 into the ACP terminal, getting rid of 1 carbon. So here we are. Then using the beta ketoacyl ACP reductase, we reduce the keto group on the ACP terminal using NADPH. And here we have the hydroxy group. Then using the free hydroxyacyl dehydratase, we dehydrate this part of the molecule. We get rid of OH and another hydrogen here. We create a water, of course, and a double bond is created. And then we use the enoyl ACP reductase to get rid of this double bond here of the enoyl. Using, once and again, when we talk about reduction, when you hear reductase, NADPH is probably plain. And then the double bond is gone. And here we have a 6-carbon molecule. And then if we use the thioesterase, we simply free that fatty acid. And this cycle can repeat several times until we want. I mean, because this can go even through 16, 18, 20, 22, whatever, how many carbons you want. And we need, here is the recap, fatty acid synthesis. We need 4 carbons to start. Each cycle adds 2 carbons, and this is a little formula to know how many cycles actually a molecule needs, because the palmitic acid, which has 16 carbons, needs 7 cycles to be created. And this was the fatty acid synthesis. I hope you liked the video. Comment, subscribe if you want, and hope you enjoyed.